Qu'est-ce que la thérapie cellulaire CAR-T en bref?
The cell therapy using CAR T cells is a new approach to T cell immunotherapy, in which lymphocytes, T cells, are engineered using presently viral vectors to express a new receptor, a new surface protein that redirects the cells to tumor. This builds on a large knowledge whereby many investigators found that the body's immune response actually recognizes tumor. And this is mediated largely by T cells, either CD4 or CD8, and these have the ability to recognize and kill tumor. To reproduce this artificially, one takes peripheral blood lymphocytes, which have specificity against a myriad of antigens, and transduces them very efficiently. Up to 60, 70, 80% of cells get transduced with a single receptor, which then redirects all these lymphocytes against tumor. So it's a approach by which, for the first time in human history, one can reprogram the immune system to organize it with a massive attack against cancer. Now, CAR T cells bring additional innovations because they use synthetic biology, meaning new molecules. The extracellular domain of this molecule is actually a component of an antibody, usually, but it can be other moieties. Or modules. This antibody binds to a cell surface molecule expressed by the tumor. So one moves away completely from the dependence on the MHC, and so it's a universal therapy that can be applied to any patient with that particular tumor that expresses that particular antigen. In addition, the CAR Ts on the molecule that is being introduced to the T cells has an intracellular domain, a cytoplasmic domain, and that activates the T cell through two distinct pathways, providing signal 1, which replicates the natural T cell receptor, but also a second or third signal, which reproduces additional receptors, which are called co-stimulatory receptors. So the T cell receives simultaneously multiple stimulations from the single antigen, which allows the T cell to maintain a fitness against the tumor cell for multiple encounters, which it was called a serial killer. So a T cell can kill 1,000 tumor cells, one at a time, of course, but unlike T cells, which are only endowed with a T cell receptor, which kill 2, 3, 4 times and then they get exhausted, the CAR T cell can keep going, and that allows for for the therapeutic efficacy which has never been seen before.
Donc une certaine type de mémoire.
Absolutely, the memory with the CAR T cells is actually a very important component of the therapeutic efficacy, because on one hand we provide a very large number of cells at a single infusion. So the cells are prepared in the laboratory. They are infused to a patient that has been lymphodepleted, and the patient receives normally hundreds of millions or billions of cells. They provide an initial attack to the tumor, which often leads to a collapse of the tumor defenses and elimination of the tumor, and oftentimes eradication of the tumor. And we know this is likely a near complete eradication, or at least a eradication at the clinical level, because many patients remain in remission for several years. And then we talk about memory, because there is a small number of cells which continue to circulate and maintain the state of remission.
Quelles tumeurs peuvent adresser pour le moment avec cette technique?
The CAR T cells today are mostly applied to hematologic malignancies with great success. So the front runner is CD19, which obviously is expressed in T-cell leukemia, lymphoma, and so there the success rate has been quite extraordinary, especially in pediatric ALL, with a very large number of patients who actually respond, get a complete response, and with durable complete responses at 6 months, a year, and with a longer follow-up today. Behind that, right away, is multiple myeloma, which is extraordinarily promising with new targets. And I think there is now an explosive list of hematologic targets that are being tested. I think we're going to see very important results, yet improving on what we already have. Solid tumors is the next frontier for CAR T cells, and there I think the job is a lot more complex because we must identify cell surface molecules that are expressed exclusively in tumors and not in normal tissues. In the hematologic malignancies, we can afford to target lineage markers like CD19, etc. And then what happens is you eliminate the tumor, you also eliminate the B-cell compartment, but that is manageable medically. So people can live without B cells, and we know how to manage that medically, and it's reasonably fine. But when it comes to solid organs such as liver, kidney, lung, etc., it's to date has been very, very challenging to find targets that are only expressed in tumor and not in the original normal tissue or in other normal tissues. And there are examples where investigators thought to have a tumor-specific antigen, which turned out to be an illusion, because even minimal levels of expression, which are not detectable by conventional means, can actually trigger a catastrophic reaction, because the T cells have an extraordinary sensitivity. They recognize 1 or 2 molecules on a cell, and they can take the cell out. And so we have seen toxicities, also mortality for CARs that were selected based on the assumption of safety, but turned out to not be safe. So that's a very high bar, and people continue to work on that. And there are very interesting examples of targets that could be in fact safe for solid tumors. But I think it's going to be a long road. The alternative, of course, there is to develop TCR-based targeting with MHC-restricted antigens, so antigens that follow the canonical patterns of recognition between a T cell and a tumor cell. And there is a whole universe of tumor-specific peptides that are expressed on the surface of tumors, which can be targeted effectively. And this is now a work that's ongoing.
Vous avez parlé des effets secondaires. Donc, quels sont les effets secondaires de la thérapie qu'on utilise aujourd'hui pour les tumeurs d'hématologie?
So for CAR-Ts, we recognize the so-called on-target effects and off-target effects. So on-target effects is what I was talking earlier, which is the antigen that is being targeted is expressed at very high levels on the tumor, but it could be expressed also at low levels in a normal tissue, which would be targeted, let's be sure about it, by the T cells, and which will produce a very significant, even catastrophic tissue destruction. Now there are other side effects which are related simply to the hyperactivation of the T cells, and this is the cytokine release syndrome. And we have also seen neurologic complications, particularly for CD19 CARs, and this relates to the fact that a very large number of T cells going into the body very quickly, they expand into the body, they recognize large amount of antigen, and so there is a loops of hyperactivation which produce toxicity mediated by the cytokines, called cytokine release syndrome. It can be quite morbid, and it can also be lethal, and we have seen mortality, very low number, but yet we have seen mortality with cases, particularly those complicated by neurologic complication, which is for the time being largely related to the CD19.
C'est gérable, ces effets secondaires?
It is today. We have luckily identified therapeutic molecules that are available to the clinician. They were used before in autoimmunity, and they have become extraordinarily useful in the management of patients also with cytokine release syndrome. IL-6, for example, is a very important molecule. Neutralizing antibodies like tocilizumab, etc., have now become a part of the practice, and they are adopted quite early in the management of these patients. So for patients who present with complications with an early fever or early neurologic complication, tocilizumab administration can attenuate largely and prevent these very important side effects. There is also now evidence that drugs that attenuate T-cell signaling can have also a very important effect on attenuating the toxicity without preventing the efficacy of CAR T-cells. So I think we have very good news overall.
Quelle est l'importance de cette thérapie pour le CHUV? Est-ce qu'on traite des clients ici au CHUV avec la thérapie cellulaire CAR?
So we are starting. We are going to start with CAR T cells here, both commercial, because now, of course, CAR T19 is commercially available in Switzerland. So we will be soon able to give those to our patients this year. We're also looking forward to open clinical trials with CAR T cells, both commercial as well as academic. What we are working on, especially on the academic side here, is an alternative approach, which are TIL therapy. And so the TIL, unlike CAR Ts, rely on a natural recognition. So we actually rely on the natural T-cell response that is present already in tumors. So we harvest these T-cells in the laboratory, we expand them, we activate them, and then give them back to the patient. The preparation of the patient is very similar. So we give lymphodepleting chemotherapy and then infuse the T-cells, and we follow that with a few doses of high-dose interleukin-2 to support the expansion of the TILs. TIL stands for tumor-infiltrating lymphocytes. So it's a natural T-cell. Remarkably, toxicity with TILs has never been a very serious issue. Of course, there are toxicities. There are almost never toxicities related to recognition of T-cells of the antigen elsewhere in the body, so on-target toxicities, probably because these have already been naturally selected to recognize tumor. For the majority, we don't know what they recognize, and that's a big area of investigation now to understand what do these T-cells find in the tumor, which we can then use therapeutically. But at the moment, we know that once you can expand T-cells out of tumors successfully, you can give them back to patients. A proportion of these patients will benefit. And this has been largely tested internationally in melanoma. So we are actually having also the same clinical trial here we open, and we have manufactured TILs for melanoma very successfully, and we have given to patients very successfully. And these are patients who have failed all other therapeutic opportunities, including immunotherapy with drugs. So we know it works, and it also works for patients who failed PD-1 blockade, CTLA-4 blockade, etc. Now, the big opportunity here, of course, is expanding TIL therapy beyond melanoma in solid tumors. So we and others, when I was at Penn, my laboratory demonstrated a spontaneous immune response in gynecologic cancers. Others have demonstrated in other tumor types. Therefore, we hypothesize since the early 2000s that TILs in solid tumors should work. And so now we put this finally into the test. So we and others, and there are very encouraging data coming out from other tumor types, including cervical cancer, head and neck cancer, lung cancer, etc. And so we have now here opened the clinical trial for so-called all-comers. So any type of solid tumors, provided these tumors are convincing to us that can grow out good T cells, and then we were going to treat these patients and see how well we can reproduce the good results that we're having in melanoma.
Donc, c'est aussi possible pour des médecins hors du CHUV de verser des clients pour ces études cliniques?
We welcome all patients, in fact, and we are receiving patients from elsewhere in Switzerland, from Chur, from Zurich, Geneva, etc., colleagues around the country. And so we are obviously welcoming all these patients. We also see some international patients who come through. We have to screen them obviously very carefully, and it's for us the patient. We have obviously a very good criteria based on which we can accept or reject the patient for eligibility for a clinical trial. These are very well defined, and this is what the clinicians are using for putting the patients on.
Vous avez adressé la coopération en Suisse, donc est-ce que vous pourriez un peu parler sur la coopération en Suisse? On a lu sur le SAKK cette formation de recherche. Est-ce que vous pourriez un peu parler sur ça?
Yes, so in Switzerland we are now launching a very, very important initiative to develop a platform for academic T-cell therapy cooperation. The idea is that we are behind at an international level. We're a small country, but we're a country with very important talent, with very deep resources, both technological and clinical, exceptional clinicians, exceptional universities. So I think this can really put together a highly competitive program for T-cell therapy if we put ourselves together to work together, and this is what we propose to do with SAKK, and we have now formed a working group for cell therapy within the SAKK. The goal of this group is to advance clinical research and advance T-cell technology in the country, with the participation of all the university centers and all the colleagues from the non-university centers who are part of the SAKK anyway, with manufacturing of cells in university manufacturing facilities, with spreading the know-how for this manufacturing and for treating the patients across the nation, and with the ability to really recruit much faster, but also putting together the know-how from everyone, the technology from everyone, to put Switzerland, in fact, on the international scene as a very competitive program. And our goal finally is to advance technology from the laboratories into early clinical testing, to then screen these technologies to see which of these technologies, of these approaches are important and promising, and then find partnerships with our commercial partners, with pharma, who know then how to take these technologies to the next step, because our competencies are in developing early concepts, but not in distributing widely, and this is what pharma can do best. And I think there is an ecosystem for very strong partnership between and collaboration between the two partners, the academic and the commercial. I am a very strong believer in this collaboration, and I believe that we have the ingredients here in Switzerland to be extraordinarily successful.
Est-ce qu'il y a aussi un rôle de l'État comme, disons, un support financier de l'État?
So we actually have built all this with financial support from the Canton de Vaud, which has been extraordinarily generous in supporting us, knowing that in fact this initially takes an important investment, but this investment catalyzes a series of reactions that are extraordinarily productive in terms of return of investment, both in terms of public health, because in fact, you know, we can hopefully provide new therapies that will provide solutions to patients. And you know, what is extraordinary about T-cell therapy is that these therapies can be in fact curative, but the value is also beyond that, is creation of business opportunities for the community, partnerships with biotechs, partnerships with pharmas, so an economic also valorization of this platform, and also from the scientific standpoint, the opportunities that were afforded are quite amazing in terms of collaborating with our bioengineer colleagues, for example, at the engineering schools, and our scientists at the universities and the hospitals. So it's really a galvanizing factor, and I think it's worked quite well so far. Clearly then the state has a very important role in these initial investments. I hope this will continue, and I hope this will be also endorsed at the federal level, you know, an area where we actually need very important support in countries that are highly competitive at the international level. You see that there is a very strong federal or national support system for funding cancer initiatives through national cancer centers, and you see this in the US, you see this in the UK. If you think about the major advances that have been made today, scientifically and clinically, which produced major impact in the lives of patients, with the mortality declining, early diagnosis and all that, these are due to the massive investments that the governments have put in the UK, in the US, and in large countries of this sort. So federal investment in cancer are extraordinarily important. They have massive returns for the population, benefit for health, energizing the universities and the technology and the biotech sector. So I think these are well overdue also in Switzerland. We hope to engage the federal government at some point, and I think the universities themselves have also an opportunity here. I think we've seen this very successful initiatives in Canton Vaud. Hopefully there will be similar investments in initiatives in other cantons where there are very important universities across Switzerland.
Pour finir, est-ce que vous avez encore un take-home message?
For us, what is very important now are these important partnerships. We work incessantly for the benefit of our patients. And we feel a little, if you will, underpowered in this massive effort. The war against cancer is infinite. The resources required to produce new technologies are very, very substantial. And although the states, the cantons and the federal government are putting significant resources towards the university systems, towards the hospital system, it's not enough. If you look at the operating budget of cancer centers, of large cancer centers and universities and hospitals in the US, for example, 50% of that budget comes from philanthropic support. And these operational budgets are huge. And these universities raise hundreds of millions of dollars through philanthropy every year. Something that in Europe is not as common. In UK, you see that a lot. There are other countries that are now picking up, Germany, the Netherlands. I wish we can energize also the Swiss population. There is an enormous amount of wellness in this country and of goodwill, if you will. And the fight against cancer will require their support, will require the resources, and will require the generosity so that we can go up faster and more effectively towards curing cancer.
Ευχαριστώ πάρα πολύ.
Thank you.